So they say we may have nanoplastics in our arteries, and what's more, they may be killing us. This has been reported all over the place. I picked this article from NBC News, but the messaging is generally the same throughout. These 150 patients where they found plastic particles had 20% chance to have a heart attack, stroke, or death. And the ones without these particles were only 7.5%. So this is a significant difference, and the claim is that there's a correlation with micro and nanoplastics found in the arteries of the patients. Okay, this is a scientific article published in New England Journal of Medicine, March of 2024. And as usual, let's go to PowerPoint now just for simplicity. So as you may know, rather than jumping to interpretations about what micro and nanoplastics in our body mean for our health, I prefer to start by trying to determine whether those micro and nanoplastics really are in our body in the first place. So to that end, the most important things are that the only two types of plastics found, according to the authors, are polyethylene and polyvinylchloride. And the methods they use to detect these are pyrolysis gas chromatography mass spectrometry, which I will call pyrolysis GC mass spec, and stable isotope analysis and electron microscopy were also used as auxiliary detections. But this pyrolysis GC mass spec is the workhorse of the study. And so let's go over that in some detail. Okay, here it is, pyrolysis GC mass spec. Let's start in the middle. Gas chromatography, this is used quite often, and if you're a big fan of the movie My Cousin Vinny, you might remember this scene.
I used a Hewlett-Packard 5710A dual-column gas chromatograph with flame analyzation detectors.
Uh-huh. Is that thing turbocharged?
Only on the floor model.
Okay, so that was expert witness George Wilbur explaining how he determined what kind of tires were at the scene of the crime based on gas chromatographic analysis of the tire track. Gas chromatography is used all over the place, and what it really does is it separates stuff. This is a gas chromatogram of sweet orange essential oil, and sweet orange essential oil is a mixture of several different molecular compounds. And if you use GC, you can separate that out and you can see all of these different peaks. Now, George Wilbur used flame analyzation detection, which can give you the information that you see here, but a lot of the modern instruments use more advanced detectors such as mass spectrometry. And what this allows you to do is analyze and identify what these different components are as they come out. So rather than just getting all these peaks at different retention times, you're able to assign these peaks to molecular structures based on this mass spectrometry analysis. So this is GC mass spec is a workhorse for a lot of industries. It's used in synthesis labs, it's used in analytical labs, it's used in crime labs. You may see it mentioned on CSI, for example. But one place it's not very useful is an analysis of plastics. And the reason why is this word right here, gas. So this is gas chromatography, and you can't boil polymers. They're too big. So plastics are made of very large molecules that simply won't boil to get into the gas phase to do gas chromatography. So what do? Well, somebody came up with the idea of let's just not boil them, but heat them so hot that rather than boiling, they fall apart. Okay, so that's what pyrolysis is. Essentially is you heat these molecules to such a high temperature that the molecules themselves start to fall apart and they turn into smaller molecules that can be analyzed by GC mass spec. Let's go through an example. Okay, so this is polyethylene. In reality, it's going to have some branching, some side chains, but for the purposes of this discussion, let's just assume it's this long linear hydrocarbon chain, which is essentially what it is. So each chain is going to have maybe a few thousand CH2 groups. That will vary, of course, depending on the grade of polyethylene you're using, but that's a rough approximation. And then in this analysis, they used, I think you typically use maybe a milligram or so, which might be a few thousand trillion chains. Okay, so you literally have about 1,000,000 trillion carbon atoms in this experiment when you do it. And then you hit it with a bunch of energy and it just blasts it apart. So what used to be this really long chain of a few thousand CH2 groups breaks into these smaller fragments like the one I've shown you. And of course, it's not particularly specific. You get a bunch of different chain lengths. A lot of them are these alpha omega dienes. That means double bonds at each end of the chain. And it's just this big mixture of fragments. So because it's so complex, what is typically done in pyrolysis GC-mass spec of polymers is you pick one to be your indicator fragment, okay? And in the case of polyethylene, one that's commonly used is this big molecule at the top called 1,20-hene icosadiene. And it's just a 21 carbon diene with a double bond at each end. And you can see here in the supplemental of the scientific paper that this is in fact the indicator molecule that they used. But the thing that's really important to remember about this is a molecular piece of a polymer. So this is not a polymer that's being observed directly. It's a piece. So it's kind of like finding one Lego piece from a set. So you can look at that piece and you might recognize it as being from the Death Star set or something else. But you really just found one piece. And if you only have a couple Lego sets in your house, maybe you can tell for sure that that piece is from the Death Star set. So let's talk a little bit more about this polyethylene indicator fragment. Okay, this 1,20-hene icosadiene. In order to analyze for that, it's really important to isolate the micro and nanoplastics from everything else, which we'll call the matrix, before analysis. Why is that? Well, because anything that you don't remove gets blasted, gets pyrolyzed right along with the micro and nanoplastics, if there were any in there in the first place. And anything that gets blasted that has a long hydrocarbon chain, so more than 21 CH2 groups in it, is expected to form this PE indicator. So if you have anything in, say, an arterial plaque sample that has a greater than 21 carbon chain in it, when you pyrolyze this mixture, it will make this same polyethylene indicator molecule. And the thing is, this analysis cannot distinguish polyethylene-derived 1,20-hene icosadiene from any other kind. That is, if you have something else in there that's making this nominally PE indicator, you will think it's polyethylene because the piece is identical, okay? So that's not to say that these matrix elements are identical to polyethylene, but the molecular fragment that gets generated from the pyrolysis is identical. So there's absolutely no way to know whether this is coming from polyethylene or it's coming from something else that was in your matrix that you didn't remove. So it's a little like finding that same Lego piece in the middle of the mall, okay? And now you can't be quite so certain it was from the Death Star because Lego pieces are used in all kinds of different Lego sets, and you could maybe have the exact same piece that's used in some other Lego set. Okay, so what could possibly go wrong? What else has these long hydrocarbon chains that might be left over after doing your isolation procedure of your micro and nanoplastics? Well, lipids. So these are fats, essentially. They come in many different forms. But they're a lot of our body. So we're maybe 15% of the body is lipids. which you compare that to a lifetime of microplastic intake, which is about 0.0001 weight percent of the human body. So you need roughly 99.999% removal of these lipids. Anything less than that, and you're going to have a significant amount of lipid there relative to your micro and nanoplastic content. Now, the good news is that most lipids have shorter chains. And the shorter chains, so if they're less than 21 carbons, it's not a problem because you can't blow something apart that's less than 21 carbons and make something that's 21 carbons. So it'll only get smaller. But there are a few percent of these lipids that have hydrocarbon chains that are greater than carbon 21. They're called very long chain fatty acids for obvious reasons. And if you factor that in, you really only need about 99.9% of your lipids to be removed in order to have a chance of seeing your micro and nanoplastics. The bad news is that these are the ones that are really hard to remove. So because the way this is usually done is they generate the carboxylate salt, and those are generally soluble in water. So they can be filtered off or centrifuged to remove the liquid, the aqueous liquid from the top, and in theory isolate your micro and nanoplastics. The problem is when the chains are really long, they're not as hydrophilic, so you have enough of a hydrophobic group on there that the solubility isn't very good anymore. And in fact, these are surfactants, okay? So they're kind of like soaps, and they can do all kinds of things to your phase complexity in an aqueous solution. And an additional complication, unfortunately, is that if you have any calcium or magnesium in there, which are obviously present in a lot of the human body, they can form precipitates with these longer chain carboxylates. This is essentially the same phenomenon as soap scum. Okay, so if you have these long chain carboxylates and you have some calcium in there, it will form soap scum. So it's really challenging to remove these the lipids, especially the ones that are the most important to remove, which are the very long chain ones. So what else could be in there? Well, waxes. So these are blends of long chain alkanes, esters, alcohols, and acids. For example, candelilla wax. This is 50% hydrocarbon. Okay, so you're talking about something with a chemical formula that you see here. You might recognize that that's exactly the same as the simplified version that we used for polyethylene. It's just that the number of carbons is more around 30 as opposed to around 1,000. But chemically, it's essentially identical. Now, candelilla wax is generally regarded as safe. So I couldn't find any dietary uptakes on an annual basis. I don't know what the average uptake is. It is a common food additive. It's in things like chewing gum, fruit coating, chocolate. I did find that a typical chocolate bar, just a normal sized chocolate bar, has about 66 milligrams of these hydrocarbons from the added wax to that chocolate bar. This is about a lifetime of microplastics intake. Okay, so you're talking about something that is semi-crystalline, just like polyethylene, that is chemically nearly identical to polyethylene. So if wax and polyethylene are nearly identical, you may be asking yourself, why do we just mindlessly munch one of them and send teams of scientists to try to track down every last picogram of the other one? So another kind of wax is beeswax, carnauba wax. They are also generally regarded as safe. And then there's paraffin wax, which is a synthetic wax that it's not generally regarded as safe, but it can be FDA certified. These are all rich in greater than 21 carbon chains, and they're all commonly used in foods, especially coatings on things like nuts and fruits. So We are taking in way more of these waxes than we are micro and nanoplastics, and I don't know of any way to separate them from micro and nanoplastic polyethylene. So based on this, the method used to isolate the micro and nanoplastics from the matrix, you can see, is incredibly important. So let's look at the description in the scientific paper of how they did that. This is the description in the scientific paper of how they did that. This is all they gave us. So the usual threshold for publishing of a scientific procedure is that it enables someone else to reproduce the results. I've spent a lot of time in the lab, and I just don't know what this means. Pre-treated, I mean, was this peroxide, base, acid? Was it all water? Was there a buffer in there? I don't know. I mean, was it centrifuged? Was it filtered? There's no information here that would allow anyone to reproduce these results. And I don't know, based on the way the rest of the paper was written, I think this may have been more of a lack of appreciation for how important this isolation procedure was than, you know, a general lack of detail that was reported. But this is just, I don't know how it gets published with this. I mean, I guess this is good enough for the New England Journal of Medicine, but you know, the journals that I normally publish, I mean, you would not be able to publish this in the journal Macromolecules, for example, because it just doesn't meet the standard for publication. It doesn't allow anyone to reproduce the results. Okay, that was polyethylene. Let's talk a little bit about polyvinylchloride, the other plastic that they claim to have found. The indicator ion for this is naphthalene. That seems a little strange, right? Because polyvinylchloride has a lot of chloride on it. But when you pyrolyze that, the first thing that happens is hydrochloric acid comes spitting off, and you get these unsaturated hydrocarbons that form all these aromatic species, just sort of out of desperation, I suppose. And so naphthalene is the one they use to identify PVC. So this is also not quite so simple. Where else might you get naphthalene in your body? Well, it's a common product in combustion, so it comes from incomplete combustion. Complete combustion, you go all the way to carbon dioxide. But unfortunately, you don't always get complete combustion. And so naphthalene and a lot of other polyaromatic hydrocarbons are very common in tailpipe emissions, smoke. Naphthalene itself is reportedly a few weight percent of diesel emissions. So if you inhale some diesel particulates that come from tailpipes and that gets entrained in your blood somehow, and that gets stuck in an arterial plaque, that will present as polyvinylchloride in a pyrolysis GC mass spec analysis. There are other constituents like naphthenic acids that would generate naphthalene when pyrolyzed. And then, naphthalene was originally used as mothballs. There are other things that are used now, but it is still used as a deodorizer, repellent. And so average concentrations just in air that we normally breathe, are around the same in terms of micrograms per cubic meter as reports that I've seen for microplastics. And bear in mind that if you use naphthalene as an indicator for PVC, you're multiplying that by some factor to account for the fact that not all PVC turns into naphthalene. So a tiny, tiny bit of naphthalene could be interpreted as a significant amount of PVC. So PVC, like polyethylene, has some significant complications that really make it impossible to say with any certainty that they really were seeing PVC and not something else. And perhaps that's the reason why this very recent paper, that is really the first thorough method validation paper that I've seen in this field, they report pyrolysis GC mass spec is currently not a suitable analysis method for PE and PVC in biological matrices. So the authors of this paper, not the main one I'm talking about, but this paper that's quoted here, finally are doing the method validation and the research that really should have been required before applying this to any sort of a field sample. And I guess the analytical chemists have finally entered the chat on this, but unfortunately, there are a lot of papers out there that use this method to look at human tissues, and they don't take into account the fact that it's not suitable for finding polyethylene and polyvinylchloride, and all of the other polymers have their issues too. They're just perhaps a little more subtle or less elaborated than these. So just to summarize, what is identified in this paper as polyethylene is more likely fat. What is identified as PVC is more likely smoke. And any other study of biological tissues that relies on this method is going to have the same complications, and they generally don't address them. Okay, I just want to quickly go through the other methods they use. The stable isotope analysis, because the microplastics were identified using a technique that's not reliable for these microplastics, you kind of have to put this in quotes, okay? But whatever it is that they found in there does seem to correlate with old carbon. That's basically what this means, is it's kind of like carbon dating, that the ones that presented as having microplastics also presented as having older carbon on average, so petroleum-based carbon. And they're correct. I mean, this could indicate microplastics, but it could also be some other things we were talking about, like synthetic waxes, tailpipe emissions, because you're burning fossil fuels. Fossil fuels are old carbon. Other synthetic food additives are also based on petroleum products. So this is consistent with microplastics, but it's certainly not standalone evidence for microplastics. Okay, electron microscopy. This is a sample of plaque, and they picked out an area here in blue where they see a particle, so that white stuff behind it is where they identified a particle. And then for comparison, they have this red area where they see no particle. So in the red area, they find chloride at about 3 mass percent. In the blue area, it's higher at about 4.7 mass percent. So yeah, 4.7 is higher than 3, but I am a little cautious to interpret this, because in this red one, they also didn't find any calcium, whereas in the blue one, you start to see a little bit of calcium. So, you know, this slight difference in chloride seems like it's kind of balanced by an additional amount of calcium. So, you know, maybe that's just a little bit of calcium chloride that got seeded on that particle. And then keep in mind that PVC is about 57 weight percent chloride. So, you know, if this was really a PVC particle, it seems like we might expect a little more dramatic difference in the chloride content. So again, you know, this is supporting evidence perhaps, but as a standalone method, I don't think anyone would say that this is proof that that's a PVC particle. You know, this is a tragedy. I mean, this was a beautiful paper. It really was. And I mean, this was a big study, and as far as I can tell, everything was done perfectly, except the key analysis just isn't valid in this kind of system. And so I appreciate the pain of going through a big study like this, thinking you have an answer, thinking you've got it all figured out, and then finding out later that you really don't. And I think this kind of psychic pain of, you know, realizing that some published papers are really not valid is going to sort of permeate through this system, since that careful study finally did the method validation, some of the method validation that needs to be done for this method. And look, I've said it before, I don't like making these videos. I really would prefer, I want everyone to get it right. I really do. I don't like seeing this kind of beautiful research be tainted by use of a method that just doesn't work. So I hope that these scientists will reevaluate their data, reevaluate their method, and maybe figure out what they really were measuring, and come back with some useful information in the end. Anyway. But I wanted to point out, look, I looked at the author affiliation page in addition to the authors, and there's a lot of hospitals and research institutions represented here. But what I didn't see is any representation from chemistry, analytical science, polymer science, material science. And would that have made a difference? Well, maybe. I mean, if you had an analytical scientist on your team, they might tell you something along the lines of, you really should characterize these matrix residuals. So for example, after you do your digestion and you only have a little bit of residual material left, which you're assigning as being micro and nanoplastics, you could analyze that with things like GC mass spec and maybe HPLC mass spec without doing the pyrolysis. So look at your small molecules that are still present, and that might give you a hint at what might complicate your analysis when you do the pyrolysis GC mass spec. And it might be a little bit more convenient to convert your residual long-chain fatty acids that are still there into their methyl esters, just to simplify the GC and HPLC mass spec analyses. So, a chemist might also advise that you plan for potential false positives. For example, you might expect to find some food-grade mineral oils in there. That's a mixture of thousands of different kinds of hydrocarbons, linear and branched aliphatics, aromatics, and they could easily be problematic in doing analysis for the micro nanoplastics that you want to look for. Lubricants as well. So something like 60% of the U.S. diet is processed foods. When you do processes, you're going to use lubricants. Now, there are food-grade lubricants that are safe, but the FDA allows up to 10 parts per million of those lubricants in the food. So these also are present probably at higher levels than micro and nanoplastics in our bodies. And if you do some preliminary work, look at analysis, including pyrolysis GC mass spec of these potential contaminants that might be an issue in your analysis, it could help. A material scientist on your team might also suggest that you understand a little bit better what you're looking for. For example, polyethylene could be any of hundreds of different structures that are commercially available, and they vary based on the number and spacing and length of short chain branches, carbon. We're talking about 1 to 7 carbon branches. Also long chain branches, which are about as long as the chain itself. Molecular weight distribution. You may want to pick, 8 or 10 of the highest volume polyethylenes and analyze those separately. As material scientists might also tell you that you should probably think about particle size effects, like what are the physical properties of your polyethylene if it's been reduced to a nanoscale. You can look up the melting point and elasticity and all these things for different kinds of polyethylene. But if it's been weathered and degraded to the point that it's now a nanoplastic, how will that affect its physical properties? I mean, is the melting point going to be depressed? Is it even possible to get to the nanoscale? And your material scientist might also warn you that as these particles degrade in size, they may also change chemically, and these changes may be significant. So another thing an analytical scientist might advise you to do is to develop fingerprints for identifications rather than just relying on a single peak as representative. Look at all of them. And that George Wilbur, who did the flame analyzation detection for the tire, almost certainly was not looking at just one peak to decide what tire it was. He was probably looking at the pattern. And again, another thing an analytical scientist on your team might advise you to do is to validate the method. So look at percent recoveries by your polymer type and maybe also by particle size. So are they getting digested when you try to remove your matrix elements? Or are they coming through? And so if you just, run some studies with just the polymer, you can get an idea for, whether your isolation methods are really even relevant for the polymer types and sizes that you're looking for. And then even more thorough is to spike in some of these plastics that you're looking for on top of the tissue that you're analyzing to make sure that they survive in the presence of the tissue sample. Your analytical scientists on your team may also suggest that you consider other methods like high temperature HPLC, which works for a lot of polymers. And this is a non-destructive method that could be coupled with IR or Raman. And then if you'll allow yourself to listen to some old retired guy in his basement, I might also advise you to think about how you set your goals. If your goal is to look for something that's not there, you might get tempted to find it anyway. And if you set your goal instead as to find out whether it's there, or even better, what is there, you may have a better chance of achieving success within the constraints of reality. And in that regard, you'll have a better chance of providing information that's really useful to society. Thanks for watching. There's more to come.